And we're going to talk a lot about many different kinds of things associated with different cell types. And then some of these things we're seeing today we'll go into more detail with later, like translation and protein synthesis with the ribosomes. And some of this will be review, right? Because it's just, parts of the cell structure and function. So I'm sure that most of you have had this in high school biology, so you probably remember a lot of this. Now, when it comes to cells, there is a theory called the cell theory that all organisms are made of cells, and the cell is the simplest collection of matter that can live. So in other words, it's the smallest unit of life. So by definition, life must be at least a single cell. And then all cells come from pre-existing cells. Now, I added a question mark, the smallest unit of life, because this is a theory. It's extremely well supported. There's tremendous evidence supporting the cell theory. But because it's a theory, it's possible in the future some new evidence will be discovered that will cause us to rethink what the definition of life is. And we have some of that evidence today in the form of giant viruses. So there are these enormous viruses like the Mimi virus or the Pithovirus. These are enormous viruses. They are as big as bacterial cells, and their genomes are larger and in some cases more complex than some bacterial cell genomes. But the giant viruses do not have cytoplasm. They don't make ATP. They do not perform transcription and translation, although they may have some of the genes necessary for translation. They don't actually perform those functions. They require host cells, right, to infect and use the host cells to make new copies of those viruses. But these viruses are enormous with very complex genomes, and that raises the question, they're so large and so complex, they're almost cell-like, but they're not true cells. And so it's possible they might want to redefine what life is based on these giant viruses. So the point of the cell theory is what we currently understand and how we currently define life, but it's possible in the future we may change the definition of what life is. And now cells are tiny, most of them. A few of them are large enough you can see with the naked eye, but they're still really, really small. And so we need help to see, these tiny little things. And that help is in the form of a microscope. And so microscopes consist of a series of lenses. What we're looking at here is a compound microscope because it has ocular lenses, objective lenses, a condenser lens, so it has multiple lenses, so we call it a compound microscope. And that magnifies the object's apparent image. It makes the image bigger so we can see it. The typical microscope that is most commonly used is called a light microscope or a bright field microscope because the object is darker against a bright field of view. So you use ordinary white light. And we have a lab that will do involving the microscope tomorrow, I believe. So magnification involves enlarging the apparent image of an object. So we're not making the object bigger, we're just making its image bigger so we can see it. Now, it's not enough to simply magnify, right, to enlarge the apparent image. We also need resolution. And resolution is the ability to distinguish 2 points as separate. So it's not enough just to magnify and zoom in and get really close. You must be able to distinguish 2 points as separate. Otherwise, it will be blurry, right? All of the objects will not be clear. And then contrast involves differences in parts of the sample. So some parts will be lighter, some parts will be darker. So there's contrast involved. So you magnify the apparent image. You also need sufficient resolution to distinguish 2 points as separate. Otherwise, they're blurry, right? They're not clear, and they appear as one object instead of two distinct objects. So when it comes to a sense of scale, the unaided eye, if your eyes are good, you can see down to about 0.5 millimeter, maybe less than 0.5 millimeter if your eyes are still good, or if you have really good glasses. Below that, we need a microscope to help us see. So most cells cannot be seen by the naked eye, although a few can. They're large enough. You can tell they're there. You can't see them clearly, but you can tell they're there. And then below that, we need something more powerful like an electron microscope. When it comes to resolution, it's the energy of the source of light that's being used. In the case of an ordinary bright field microscope, we're just using white light, and that limits us to about 1,000 times magnification and a decent resolution. If you want to increase magnification and resolution, you need to increase the kind of energy that you're using. So electrons are very high energy particles. They have much shorter wavelengths than white light does. And because of that, you get much higher magnification and resolution. Now, I have another animation to show you. So I have to share my screen again to show you this animation. And it is for a sense of scale. And my browser just locked up. It's frozen right now, so let's wait and see. I can't do anything at the moment until it unfreezes, so let's give it a minute or two and see if it'll unfreeze. So what are you guys seeing right now?
The same slide you had.
Oh, just that one slide, so you're not seeing how messed up it looks on my computer.
It looked the same.
Yeah. Well, my browser's not responding, so I might have to shut down and come back. All right, I'm going to shut this down, so just hang on. I'll come back. Okay, we'll try this again. Okay, now it's working. Everybody still with me?
Yes.
And so can we see this animation?
Yes, a coffee bean and grain, right?
Right, so we'll start off with things that you may be familiar with, like the size of a coffee bean or a grain of rice, a sesame seed, a nice 12-point font that's easy for your instructor to read. And we're going to zoom in and get closer and closer. A grain of salt, 0.5 millimeter. We can all see salt grains. Amoeba proteus, also 0.5 millimeter, so 500 micrometers, right, 0.5 millimeter. And you can see these with your eyes, if your eyes are good. And then a paramecium, we're getting less than 0.5 millimeter, but if you were looking at a culture, you could see these little things moving around in it if your eyes are good. So you can still see that, but now we're getting down to the point where you need help to see things, either a microscope or a really good magnifying glass. We have a human egg, and then a teensy tiny little sperm cell. How come the egg is so big and the sperm is so tiny? Any idea why?
Is it because we get the mitochondria from like the female?
Yep, so all the sperm does is deliver chromosomes to the egg. The egg has everything needed for a cell to be a cell. All the cytoplasm, all the organelles are all in the egg. Sperm is just a chromosome delivery system from dad. Got a skin cell, photoreceptor, a red blood cell, an X chromosome, a baker's yeast. So a baker's yeast, this is a cellular organism, a single cell fungus, an entire cell. The X chromosome is bigger than a yeast cell. Got mitochondria, E. coli. Notice that the E. coli and mitochondria are similar size and shape, and they have a lot of similarities in common. And that has to do with the fact that we believe that mitochondria and chloroplast evolved from bacteria. Anybody know what we call that theory on the origin of mitochondria and chloroplast? What do we call that theory? Anybody know? Well, we will learn about it. It's the endosymbiotic theory. Get closer and closer. Measles virus, influenza virus, HIV, a bacteriophage. So this is a virus that infects bacteria. Hepatitis, ribosome, a rhinovirus. Rhinoviruses are common causes of colds and respiratory infections. Notice that they're the same size as a ribosome, right? A ribosome is that little machine that translates to make proteins. Same size as a rhinovirus. Hemoglobin, transfer RNA, an antibody to protect us against those viruses. Phospholipids, glucose, water molecule, and finally a carbon atom. So it's just a nice little animation for a sense of scale. In terms of what we can see with our eyes and what things we can't see. So we need alternate means in order to determine if something is there. Yeah, question, Zoe?
Can like really small animals and insects like see more than we can?
No.
No.
They have more limited sight, especially insects do. I mean, they see things very differently than we do, but they do not see better than we do. Certain birds, like eagles and hawks, have much greater vision than we do. But many other animals, their vision is not all that great, and so they use other senses, like a sense of smell. But it does depend on what animal you're talking about. But, you know, we see better than many things, but other things can see better than we can. But they don't often see the same detail or the same colors in the same way that we do.
Okay, yeah, I was just wondering like if they could see like, like cells and stuff in detail. I don't know.
No. No, generally speaking, they're more limited in terms of what they can see. Like, dogs will use their sense of smell much more than their eyes because their eyes don't see as well as our eyes do. But animals that rely on their eyesight for hunting, now they do have excellent vision, right? Because they need it for hunting, right? For catching prey. But it depends. Snakes have terrible eyesight, but they have a good sense of smell, and they can also sense body heat, from their prey. So it just depends. But even though most things have eyes, they're not all the same. So light microscopes have a maximum magnification of about 1,000 times, and that's due to the wavelength of white light, right? There's not quite that much energy there, so you're limited to about 1,000 times magnification. And there are different types of microscopes that we're not going to go into in this course, but if you go on to take microbiology, you'll learn about many other kinds of microscopes. But if you're looking for internal cell structures, we can see the nucleus of many cells because the nucleus is very large. We can't see in detail the other organelles. We know that they're there because we can see little dots and things inside of cells, but we can't really determine what type of organelles they are. It's not that easy to see. We would need something like an electron microscope in order to see that interior detail. But nuclei tend to be very large, so those we can see. When it comes to electron microscopy, again, I said the electrons are very high energy particles, so very short wavelengths, so you get tremendous magnification and resolution. The two main types of electron microscopes are scanning electron microscopes and transmission electron microscopes. A scanning electron microscope actually uses an electron beam to scan the external surface of a specimen, and that produces a very beautiful three-dimensional images. Transmission electron microscopy gives us much greater magnification and resolution than a scanning electron microscope does, and you're actually looking through an ultra-thin slice of a specimen. So when I say ultra-thin, I mean super thin. Like you could slice up a protozoan or a bacterial cell at least once, if not more than one time. So extremely thin slices. And that allows us to see internal structures. Now, all of this, produces black and white images, but with computers, we can colorize them now. But here we see some cilia from a scanning electron microscope. So we see, what looks more three-dimensional here, as opposed to a slice, through the cilia. So a slice allows us to see internal detail. A scanning electron microscope scans the outer surface and produces some really cool looking images, especially when you colorize them. But we get lower magnification with the scanning electron microscope, usually around 10,000 times. A transmission electron microscope can magnify up to at least 100,000 times with very excellent resolution. Now you can go beyond the electron microscope, which we're not going to go into in this course, but we can actually magnify down to the atomic level now with the help of computers. So we can actually see how atoms are arranged in molecules now. So that's how far the technology has progressed. But when it comes to cell types, we have two major types, prokaryotic cells and eukaryotic cells. The prokaryotes comprise the domains bacteria and archaea. And then eukarya has all the eukaryotic kingdoms, protista, fungi, animals, and plants. All cells have a plasma membrane, also called a cell membrane. They're the same thing, plasma membrane, cell membrane. And then within the plasma membrane is a semi-fluid substance called the cytosol, so it's watery. Now, when it comes to the term cytosol and cytoplasm, there is a difference, although we often use the terms interchangeably. Technically, the cytosol is just a watery part, not including organelles like mitochondria, chloroplast, the nucleus, etc. When you include the organelles, you usually use the term cytoplasm. Right, so cytoplasm is everything within the plasma membrane, and the cytosol is just a watery part, minus the organelles. But sometimes they're used interchangeably, especially when it comes to prokaryotic cells, because prokaryotes don't have any organelles. There's no nucleus, there's no mitochondria, and so we typically refer to everything within the plasma membrane as the cytoplasm for a prokaryote. But for eukaryotes, we tend to distinguish the liquid part, the watery part, from the organelles. Of course, there are many chromosomes inside of eukaryotic cells. In the case of prokaryotes, they only have one circular chromosome. And all cells have ribosomes, which are the little machines for protein synthesis. So prokaryotes, no nucleus, right? The word prokaryote means pre-nucleus, so before a nucleus. The DNA is concentrated in a region in the cell called the nucleoid, but there's no membrane surrounding the chromosome in bacteria other than the cell membrane. No mitochondria, no chloroplasts, no Golgi, no endoplasmic reticulum, no membrane-bound organelles are in prokaryotic cells. And see here, we have the term cytoplasm, all right, because there's no organelles, so everything within the plasma membrane is called the cytoplasm. In general, prokaryotic cells are much smaller than eukaryotic cells. There are exceptions, but in general, they're much smaller. Most bacterial cells have a cell wall. Not all of them, but most do. Underneath the cell wall is the plasma membrane, and then you have the cytoplasm, but no organelles. And there may be outer structures like flagella, which help the bacteria to swim. Some bacteria have them, some don't. Eukaryotes do have a nucleus, which is where you find most of the DNA. Not all of it. Mitochondria and chloroplasts also have a circular chromosome, just like prokaryotes do. There are many membrane-bound organelles, and we're going to go through all of those today. And of course, the cytoplasm includes everything, including the organelles. As I said, eukaryotes tend to be larger, but there are exceptions. So the plasma membrane, we'll talk about that tomorrow, but that will not be on the exam. So plasma membrane is not on the first exam. But it is made of phospholipids, right? That a lipid bilayer. And it's a selective barrier, so it's selectively permeable. Allows certain things to diffuse through it, like oxygen, certain nutrients, certain wastes. It depends. And it's made of phospholipids, as we talked about last time. So here we see a plasma membrane, right, the lipid bilayer. We have proteins embedded in the bilayer. Some proteins will be attached to the outer surface, some may be attached to the inner surface, and some go all the way through. We'll talk a lot more about those proteins tomorrow. Now, since diffusion is important for things like oxygen, size is a limiting factor when it comes to cells. And we talk about surface area relative to the volume, or the surface to volume ratio. So this limits the size of cells. As the surface increases by a factor of n squared, the volume increases by a factor of n cubed. So as you increase the outer surface area, the internal volume increases much more. So this creates a problem for diffusion because molecules need to be able to diffuse all the way to the center of the cell. And if a cell is too large, that doesn't happen efficiently, so that those cells would not be able to survive. Small cells have a much greater surface area relative to the volume. That helps with diffusion. So here we're going to look at an example. The math is not complicated, but so don't worry about the math here. We're not going to use any units because the units don't matter. We're just trying to compare the numbers to see, right? Because ideally what we want is a large surface to volume ratio. That allows for efficient diffusion. If the surface to volume ratio is small, that's much harder for diffusion to occur, so that's not ideal. So in the first example here, we have a little cube. Remember, cubes have 6 sides. So this is our, example of a cell type, a cube. And when it comes to surface area, it's the sum of all the surface areas. And remember, in a cube, the surface area of one of the sides is the height times the width. And since our size is just 1, it doesn't matter whether we're talking about 1 micrometer or whatever. Units don't matter here. We're just trying to compare numbers. So each side has a surface area of 1, and there are 6 sides, right? So 6 times 1 is 6. When it comes to the volume, remember volume is a length times width times height. So in this case, 1 times 1 times 1 is 1, right? So it's the volume of 1. And the surface to volume ratio is the surface area divided by the volume. 6 divided by 1 is 6. Okay, so we have a relatively large surface to volume ratio here. Now what happens if we increase the size of our cube from 1 to 5? So now each side has a measurement of 5, right? So the area for each side is 5 times 5, which is 25, and there's 6 sides, so 6 times 25 is 150. So that's the surface area on all the sides of the cube, 150. The volume, again, length times width times height, 5 times 5 times 5 is 125. So that's the volume. Surface area, 150. Volume is 125, so 150 divided by 125 is 1.2. A much, much lower surface to volume ratio. That's not good. You want a high surface to volume ratio because that allows for maximum diffusion to occur. So increasing the cell size is not advantageous. It creates a problem for diffusion. So what if we put a bunch of little cells into that same volume? So we've got the same volume here, but now we've got a bunch of little cells. So if you add up all the surface areas of all these cells, that comes out to 750. If you calculate the volume, it's the same volume, 125, because it's the overall same dimensions, right? A single cube or a bunch of little cubes, it's the same volume. But notice when we divide our surface area by the volume, again, we have that high surface to volume ratio of 6. So if your cells are small, you can pack a lot of them into the same volume, and you still have that great surface to volume ratio, as opposed to just having one large cell. And so that's why cells are small, and that's why your tissues and organs are composed of many, many little cells, right? Because you still have a high surface to volume ratio, even though you're packing a lot of cells into that same volume. And so the main point here is that surface to volume ratio limits the size of cells. As you increase the surface area, the volume increases a lot more. And that keeps cells small. Now, there are some exceptions, but are there any questions about surface to volume ratio? Again, it's all about diffusion. You want to have a large surface area relative to the volume. Yeah, question? So, how do you deal with trying to be big, right? Because there are some cells that are big. Oh, one way is to make your cell rod-shaped. In that way, you can get your surface to volume ratio such that diffusion is easy if you have a rod, versus a big round cell, that's too hard for stuff to diffuse in and out of. But if you squish it and make it a rod, that makes diffusion much easier. And there are some large rods, some large bacterial rods. There's one that is larger than paramecia, right? Paramecia are eukaryotes, but this was a prokaryote. It was enormous. You could see it with the naked eye. And when scientists first saw it, they just assumed that it was a protozoan and kind of ignored it. When someone looked more closely at it, they realized there's no nucleus in here, no mitochondria, no Golgi, no ER, so there's no organelles. It was a prokaryote, but it was huge, enormous, but it was rod-shaped. And that's how it could get away with being so huge. Likewise, there's a bacterial cell, which is round, but it's got their microphone on. So it's round, so it's huge, and it's about 3/4 of a millimeter in size, so you can see that with your eyes. But it's a prokaryote, and this particular organism, what it has is a giant storage granule in the center, right? So it's got this huge storage granule, in this case with sulfur, and so its cytoplasm is spread very thinly all around that huge storage granule. And that's how it gets around the surface to volume ratio, because the cytoplasm is spread very thinly around that huge storage granule. So that can allow that cell to be large, but in fact, most of the volume is just for storage of sulfur. Red blood cells. If you look at a red blood cell, you know that it has a double concave shape, right? So it's not round, it's not spherical, it's double concave. It's as if you took a sphere and you pushed in on the sides and squished it. That's how your red blood cells get around the surface to volume ratio problem. It's because the sides are squished inward and you get that double concave shape. So there are ways to try and get around the surface to volume ratio, problem for cells. So there are some modified cells which can exist and be larger than they should be, because either the sides are squished in, or they're rod-shaped, or the cytoplasm is spread very thinly over a storage granule. So there are ways to deal with this problem, but the surface to volume ratio limits the size that cells can be. So eukaryotes have many membranes within the cell. So of course you got the plasma membrane, which surrounds all the cytoplasm, but you got a lot of membrane inside the cell because the nucleus is surrounded by membranes. Mitochondria, chloroplasts, the Golgi, the endoplasmic reticulum, they're all membranes. So there's a lot of membrane inside the cell as well. When it comes to plant and animal cells, they have mostly the same internal structures, mostly the same organelles. Here we see a typical animal cell. Notice all this folded membrane in here. So there's a lot of membrane associated with the Golgi and the endoplasmic reticulum. Even the nucleus has 2 layers of membrane at the nuclear membrane. So we got lots of membranes. Mitochondria have 2 membranes. So there's a lot of membrane inside the cell. And there may be some outer structures on the cell as well, depending upon what kind of cell it is. It may have a flagellum, it may have cilia. It depends on what kind of eukaryotic cell we're talking about. A plant cell has almost all the same things inside, except it also has chloroplasts, which also have 2 sets of membranes in there. There may be a vacuole, which can store food like starch. It can store some other nutrients and things inside of it. And then, of course, a plant cell does have a cell wall. And what's a plant cell wall made out of? Cellulose, right? And so even though there are these rigid cell walls in between cells, there are ways that cells can communicate with each other, like the plasmodesmata. So there are connections in between the cells that have cell walls, so that way they can still talk to each other and exchange information. As I mentioned, the nucleus contains most of the DNA inside the cell, but there is a circular chromosome inside the mitochondria and inside chloroplasts. Ribosomes are little machines used for protein synthesis. So the nucleus does have a nuclear membrane called the nuclear envelope. It does have pores in the nuclear membrane so that things can move into and out of the nucleus, especially messenger RNA molecules that are transcribed in the nucleus but must then be transported to the cytoplasm where they can hook up with a ribosome so protein synthesis can occur. So here we see, this is called a freeze fracture image. So they actually took the specimen, froze it, and then they broke the ice, and that ripped away some of the membrane, right, the outer membrane. And so you can see the inner membrane, and you can see the pores in the membrane. So lots of pores, right, to move stuff into and out of the nucleus, especially messenger RNAs, which must be transported to the cytoplasm, where they hook up with a ribosome. It could be what's called a free ribosome that's floating around in the cytosol, or it could be ribosomes attached to the endoplasmic reticulum. In that case, the protein, or initially it's called a polypeptide, as it's synthesized, it moves into the endoplasmic reticulum, where it will be further modified and then transported to wherever it needs to go inside of a cell. So the DNA and the proteins associated with the DNA is called chromatin. So eukaryotes have these very long linear chromosomes, so they're very stringy. And a way to help organize, all that stringy DNA is to wrap it around proteins, and that helps to keep it organized. When the DNA condenses, we see visible structures that we think of as chromosomes. So when we do mitosis, we'll actually look at condensed chromosomes. The nucleolus is located in the nucleus, and it's the site of ribosomal RNA synthesis. So ribosomal RNA is a structural component of ribosomes. Ribosomes are made of ribosomal RNA and protein. They perform protein synthesis, also known as translation, where the messenger RNA is read and translated into protein. I already mentioned free ribosomes floating around in the cytosol, and the ones that are attached to the endoplasmic reticulum are bound ribosomes. So here we see endoplasmic reticulum, and you can see the black ribosomes that are stuck to it. So the rough ER. We also have some free ribosomes floating around. Ribosomes are composed of two units, the large and the small subunit. They normally only associate to form a ribosome when protein synthesis is occurring. And then they break apart into the large and small subunits. So there is all this membrane, right? The nuclear membrane, the endoplasmic reticulum, and also the Golgi. We have membrane-bound organelles like lysosomes and vacuoles. And so we call this the endomembrane system, because a lot of the proteins that are synthesized are transported through the endoplasmic reticulum to the Golgi, and from the Golgi to the plasma membrane, where they can be secreted or perhaps embedded in the membrane. So we have this endomembrane system. And so vesicles are little bubbles, like little fat bubbles of membrane, which contain the proteins that have been synthesized. And so they bud from the endoplasmic reticulum, and they move to the Golgi, where they fuse with the Golgi. And you can think of the Golgi as like the post office, which will then deliver or distribute those proteins to wherever they need to go. So more vesicles bud off from the Golgi, and then they go to wherever they're needed. If they're needed to be secreted, then that membrane-bound vesicle will fuse with the plasma membrane to release those contents to the exterior. So there's a lot of membrane inside of eukaryotic cells. So the endoplasmic reticulum accounts for more than half of the total membrane inside of the cell, and the ER membrane is continuous with the nuclear envelope. So it's all membrane, and the ER touches and connects to the nuclear envelope. There are two types of endoplasmic reticulum, smooth and rough. Smooth has no ribosomes attached to it. The rough ER does. And that's just how it looked in the electron microscope. Right? Smooth ER had no ribosomes, but the rough ER was covered with ribosomes, so it looked rougher. And that's how they named it. So the nuclear envelope here, it is continuous with the endoplasmic reticulum. As I said, proteins get synthesized and moved into the ER, and from there they'll be modified. A vesicle will bud off and head for the Golgi so that the Golgi can deliver wherever those proteins need to go. But we do have smooth ER and rough ER. Smooth ER is involved in lipid synthesis. So it helps to synthesize all the lipids needed by the cell. It also metabolizes carbohydrates. It contains enzymes to break down cellular poisons, and also stores some calcium, right, for use inside the cell. The rough ER has the ribosomes attached to it, and they secrete glycoproteins. So these are proteins that are synthesized, moved into the ER, and then they're modified by having sugars added onto them. So a glycoprotein has a sugar attached to the protein molecule. And then vesicles will bud off the ER to head to the Golgi, right, so that they can be delivered to wherever they need to go. So the rough ER also does synthesize a membrane. So the Golgi, I mentioned the Golgi apparatus several times. These are also membrane-bound sacs. We call them cisternae. And the functions of the Golgi is to modify products from the endoplasmic reticulum. So when a vesicle buds off from the endoplasmic reticulum, it moves towards the Golgi, and the vesicle fuses with the Golgi membrane to release the contents inside the Golgi, which will be further modified, and then they'll be sorted and packaged for delivery in little transport vesicles that bud off from the Golgi. And so they may deliver their contents to the plasma membrane, right, to be secreted. They may form lysosomes, little vesicles containing all kinds of nasty things. So here we see the Golgi, right? What they call the cis face is where the vesicles from the ER will fuse with the Golgi membrane, and then those products may be modified for delivery, and then a vesicle buds off and heads for wherever it needs to go. So cells are constantly producing many, many proteins. Some of them are used inside of the cell, others are secreted to the exterior, others get embedded in the plasma membrane. So many, many different kinds of proteins. Remember last time we talked about proteins and all the different categories of proteins, enzymes, transport proteins, structural proteins. So many, many different kinds of proteins. So here we're just seeing, again, the whole process. We have the nuclear membrane, which is continuous with the endoplasmic reticulum. Vesicles will bud off from the ER, head to the Golgi, and then from the Golgi, they'll be sorted and packaged for delivery to wherever they need to go. If it's a secretory protein, it's secreted. It goes to the plasma membrane to be released. So a lot of activity happens inside the cell because there's lots of proteins being synthesized all the time. They have to be modified, packaged, sorted, and delivered all throughout the cell. So again, this is the endomembrane system. Nuclear membrane, endoplasmic reticulum, Golgi, and different kinds of membrane-bound vesicles. A lysosome is one of those membrane-bound vesicles. So it contains nasty things like hydrolytic enzymes. So these are enzymes which can digest macromolecules. Lysosomes serve different purposes. They can be used inside of a phagocyte. A phagocyte is a cell that eats other things like dead cells, bacteria, viruses, and then a lysosome has all these nasty things in it to help destroy what got swallowed by that phagocyte. Lysosomes are also known as suicide sacs. When a cell dies, the lysosomes release all their nasty compounds to help destroy that cell. I just mentioned a phagocyte, and the process is phagocytosis. Now, cells also want to move contents from the outside to the inside, like food and food, molecules, so macromolecules. A lysosome helps to break down those macromolecules into smaller pieces, which can then be used more easily by the cells. If a cell wants to eat itself, we call that autophagy. Again, the suicide sacs help to destroy that cell, or sometimes organelles like mitochondria get old, worn out, damaged, and they have to be recycled. So a lysosome will help to destroy that mitochondrion so it can be recycled. So here we see phagocytosis involving food molecules, macromolecules being, well, engulfed by that cell using a food vacuole, a membrane-bound organelle, and then the lysosome will fuse with that in order to break down those macromolecules into smaller pieces. In the case of something like a damaged mitochondrion or an old mitochondrion, a lysosome will help to destroy that, to recycle it. And the lysosomes can also kill that cell if necessary. Any questions or comments? I know this is all just pretty easy stuff. So food vacuoles are formed by phagocytosis. Technically, the process is called receptor-mediated endocytosis. It's where these food molecules attach to cell surface receptors, and the cell's like, oh, this is something good to eat, and so it engulfs, right, those food molecules. Unfortunately, that same process of receptor-mediated endocytosis has been usurped by viruses. Animal viruses have evolved to exploit that natural process of receptor-mediated endocytosis. So when the virus lands on the host cell and attaches to those cell surface receptors, the host cell thinks, oh boy, this is something good to eat. And the host cell engulfs that virus, thereby infecting itself. So animal cells infect themselves with viruses because they're tricked by those viruses. The host cell thinks it's, engulfing something good, when in fact it's engulfing a virus and infecting itself. Certain cells have contractile vacuoles. A good example is a paramecium, which lives in fresh water. So tomorrow we're going to learn about osmosis and tonicity. And the problem is, if you're a cell, your cytosol has all kinds of solutes in it, all kinds of things dissolved in it. If you're that kind of cell and you're living in fresh water, in essentially pure water, the water will tend to move into that cell, which would cause that cell to explode. And so contractile vacuoles are a way to suck up the excess water and pump it back outside. A central vacuole can be used to store all kinds of things. They're often found in plant cells. They can store water, they can store carbohydrates, sugars and starches, which the plant can use for food. Mitochondria are the powerhouses of the cell. You probably learned that in high school biology. This is where most of the ATP is formed inside of cells. And that process, aerobic cell respiration, we'll get into in a lot more detail than you want to know. Chloroplast, another way to make ATP using photosynthesis. We'll also go into that into a lot more detail than you probably want to know. But mitochondria and chloroplasts, even though they have double membranes, are not part of the endomembrane system. They have proteins made by free ribosomes, and they have their own circular chromosome, so their own DNA, but it's just one circular chromosome. In the nucleus, there are many long linear chromosomes, but in mitochondria and chloroplasts, they have a small circular chromosome. Mitochondria are in nearly all eukaryotic cells. This didn't convert that well, but it's just there's an organism which is a eukaryote, but it doesn't have any mitochondria or mitochondria-like organelles. So there are some things which have these structures called mitosomes, which are kind of like mitochondria, but not as sophisticated. But there is this eukaryote which doesn't have any mitochondria, so it can make ATP by other mechanisms, and we'll learn about that when we learn about anaerobic respiration and fermentation. So not all eukaryotes have mitochondria or mitochondria-like organelles. There's at least one example that doesn't have any, and that's kind of strange. But within the mitochondria, they do have two membranes, an outer membrane and an inner membrane. And that inner membrane is very highly folded, and we call those folds the cristae. And the reason for that highly folded membrane is that it provides a lot of surface area in a small volume. And as we will learn, that inner mitochondrial membrane has the electron transport chains needed to make ATP. And so by packing a lot of membrane into a small volume, you maximize the efficiency of the mitochondria. So here we see the inner and the outer membrane, and the inner membrane is very highly folded. Here's an electron micrograph actually showing you a cross-section of a mitochondrion. So in all this inner membrane are the electron transport chains needed to make the majority of ATP inside of a cell. And so by folding the membrane, you dramatically increase the surface area relative to that tiny volume inside the mitochondrion. And there are ribosomes inside the mitochondria, so mitochondria can synthesize their own proteins, the ones that they need inside the mitochondria. When it comes to chloroplast, another energy-producing organelle, although this time photosynthesis, and it'll be the mechanism of ATP production. Chloroplasts are a member of a family of organelles called plastids. So chloroplasts contain chlorophyll, a green pigment, and the one that absorbs light energy and uses it for photosynthesis. And we find chloroplasts in the leaves of plants, other green organs of plants, and in algae. And remember, the chlorophyll is green because that's the wavelength of light being reflected back to us. And that's why we see the green color. So again, a lot of membrane is associated with a chloroplast. You do have a double membrane, and then you have membrane-bound sacs inside. So you got a lot of membrane. And we call those sacs the thylakoid sacs or thylakoid membrane. Notice they're green because this is where the chlorophyll is. And so this is where photosynthesis is actually going to occur. There are many similarities between chloroplasts and mitochondria. They are both energy-producing organelles. Within a cell, it's not just a random mixture of things floating around. There is actually a complex network called the cytoskeleton, which is inside of cells. It's a network of fibers. And you can't see them unless you use special staining techniques. And we're going to see some pictures of that soon. But all of the cytoskeleton, it helps to organize all the cellular structures, all the organelles right within a cell. So it's not just a membrane-bound sac with a bunch of stuff floating around inside of it. That's what they used to think a cell was until they discovered the cytoskeleton. And there are three types of fibers. There are microtubules, which are made from the protein tubulin. And so the tubulin subunits can associate, and they can grow a microtubule, or they can disassociate, and the microtubule, breaks down. We have microfilaments made of actin, and there are intermediate filaments made of different kinds of proteins. But the microtubules and the microfilaments are extremely important, especially microtubules, when we learn about mitosis, we'll see just how important microtubules are. So the cytoskeleton helps to support the cell and maintain the shape of a cell. It interacts with the motor proteins. So in the video animation, we saw those things that were walking on a filament, and they were carrying something in the cell. And so they walk along these filaments and microtubules. And so the motor proteins are pretty cool. They do work a whole lot faster than what you saw in the video. But they walk along and things all throughout the cell. There's some recent evidence suggesting that the cytoskeleton may help to regulate certain biochemical activities inside of a cell as well. So once again, a cell is not just a water sack with a bunch of stuff floating around inside of it. It's actually very highly organized inside the cell. So here's one of these motor proteins, an electron micrograph showing you a motor protein, a microtubule, and vesicles that are being carried by the motor protein, right? So a vesicle, it doesn't necessarily just float around through the cell. It's actually transported by a motor protein walking along a microtubule. Motor proteins can walk forwards as well as backwards, which is something they didn't originally know, but they can actually walk backwards. They can disconnect from a microtubule. So for example, if the vesicle is big and the motor protein runs into another microtubule, which is blocking its path, it can lift off, rotate, reconnect to the microtubule and go underneath the other microtubule, just blocking its way. So motor proteins are really cool, and they didn't realize all the things that they could do, but they can move forwards and backwards. They can unhook from the microtubule and float around to the other side of the microtubule and continue to transport that vesicle somewhere. Well, motor proteins do require lots of ATP in order to move, so they need lots of energy. Exactly how the motor protein knows to go where it goes, I don't know. I'm not sure if they know exactly how that is, but the various proteins, one of the modifications is like a zip code, a molecule that is attached to them, which helps to tell the cell where to deliver it. But I don't know all the exact details, but it's not a random thing. The motor proteins move in a specific direction to transport a vesicle to someplace else inside the cell. So it really is fascinating, all the stuff that happens inside of a cell, because they used to think it was just a water sack and stuff would just kind of randomly floating around inside of it, but in fact that's not the case. Many things are being directed, right? They're being moved purposefully from one place to the other inside the cells. Now here we see all those different microtubules and filaments. If you stain them with a fluorescent stain and hit them with UV light, then all the filaments will glow and you can see how complex the network is inside of a cell. So again, it's not just a water sack. There is a complex network, a cytoskeleton inside of all cells, and that helps give them shape and structure. It creates little highways so that motor proteins can transport things from one part of the cell to the other. So a very, very complex network of these protein fibers inside of cells. Now, microtubules grow from a structure called the centrosome, which is near the nucleus. This is extremely important when it comes to mitosis And so we'll talk in a lot more detail when we get to mitosis. But the centrioles are the actual structures, and the centrosome is that region where we have all the centrioles, and the microtubules grow out from the centrioles. And as we're going to learn in mitosis, the microtubules grow out and they attach to the chromosomes, right, to push and pull them and line them up so that all the chromosomes can be divided equally among the daughter cells. But we'll learn more about that in more detail when we actually talk about mitosis later. Cilia and flagella. So some cells need to swim. Other cells are part of tissues that are trying to move something like mucus away, right, from the body. And so microtubules control the beating of cilia and flagella. And so these are locomotor structures. And so they, again, use a lot of ATP. They do form what's called a molecular motor, which allows a cilia or flagellum to beat, right, to move. And so the purpose, some things like paramecia are covered with cilia that they use to swim. Other protozoa, like Euglena, have flagella. Some bacteria also have flagella, which they use to swim. Some tissues, like our respiratory tract, the respiratory epithelial cells, are covered with cilia. And the purpose of the cilia is to move the mucus, right, out of the lungs, right? Because when you inhale, the air you breathe has particulates in it. You may have dust, maybe bacteria, if you're allergic to pollen. Right now, there's lots of tree pollen, which gets stuck in the mucus, and then the cilia move that mucus up and out of the lungs. And so that's their purpose. So we find cilia in very complex animals, and we find cilia in very simple single-celled animals like paramecia. And they're all designed and built the same way. And we mentioned this last time, that common design plan strongly suggests a common ancestor. Now, within a cell, you can see structures moving around sometimes, and we call this cytoplasmic streaming. And this helps to distribute materials within a cell. And so let's look at another little video. I will share my screen again, and we're going to look at the inside of a small plant leaf. And this is an aquatic plant called Elodea, or Elodia. And so it is an aquatic plant. It's one that we actually would have looked at in lab, and some of you would have been able to see this. So here we see the chloroplast being moved around inside the cell. And so this is cytoplasmic streaming. So the chloroplast can move depending upon where the light is, right, where the direction of the light is coming from, right, to help maximize photosynthesis. But we see the rigid cell walls, right, giving the cells a rectangular shape. We have these large green chloroplasts, which are moving around, and they will actually kind of go all the way around the perimeter and move, cyclically, within the cell. And so we call this cytoplasmic streaming. Presumably, all these little things are little organelles inside the cell, just randomly vibrating around. It could be some microorganisms on the outside of the cells that are in the water that these plants are in. It's hard to tell. So some really good cytoplasmic streaming there. So you would have been able to see these kinds of things in the lab when we played with the microscope. So we would have looked at the Eladia leaf. You guys would have gently scraped the insides of your cheeks to look at some cheek cells. So we would have done some cool stuff if we had been able to meet face to face. So that's enough of that. And it never takes me back to where I was, so we got to start over and jump around to get there again. So cell walls are something that plants and fungi and some prokaryotes have. Animal cells do not have cell walls. We do not crunch when we move, right? Our cells are just membrane. But the cell wall helps to protect those cells. It helps to maintain their shape. And when it comes to the uptake of water, a cell wall can help to protect the cell from exploding, right? Because water is moving into the cell. If it can't get the water back out fast enough, the cell will explode. If you're something like a paramecium, you can pump the water back out. But a plant cell actually needs water to move into the cell. That makes the plants very rigid, so they stand up. When it comes to prokaryotes, a cell wall helps to protect them from exploding. Oh, and so that was it. So we got through everything today. Any questions about the cell? I know it's not hard material. It's just a lot of different things, but it's not complicated material. But any questions? So I'm sure that a lot of this was review, right? So you learned about cells in biology in high school, so you remember some of the stuff. Some of these things we'll talk about in more detail later when we go into specific things like mitosis or synthesis, DNA replication. But any questions? Well, the benefit